Hi, everyone. Thanks so much for joining us today. My section, I'm going to be telling you about the CellScape platform from Bruker Spatial Biology, which is our platform for precise spatial proteomics. And so just to orient you all in terms of where this platform sits within the broader Bruker Spatial Biology portfolio, you guys are all familiar with the GeoMx Digital Spatial Profiler, really good for super high plex whole transcriptome RNA, really high plex 570 plus protein multiomics with this ability to do fast and flexible kind of um, uh, uh, transcriptomic tissue exploration in a multicellular but still spatial fashion. Um, then you're familiar with the Cosmix SMI, which has up to 6K RNA detection, up to 70 proteins with really good single cell resolution. And where the CellScape fits in here is really best in class resolution, focused on flexible, high plex protein detection around 100 or so, with a really high dynamic range and robust and straightforward assay design and flexibility. And this is the platform that I will be focusing on for this session. High level, the CellScape system is comprised of two main components. There's an imaging box on the right, pictured here on the right, and then on the left is integrated automated fluidics liquid handling to ensure walkaway automation. And high level, the way that the platform works is we're generating multiplex immunofluorescence imaging. I'll talk in a minute in a little bit more detail exactly how we generate these images. But once we have generated the multiplex image, we perform automated cell segmentation, draw a border around each individual cell, and then measure the fluorescence intensity for each marker in the assay within that defined cellular border. And from there, we have a single cell data set from which we can do phenotyping either via modern unsupervised clustering or more classical kind of hierarchical gating. And in either case, out of that, you get absolute cell counts of different populations of interest. And of course, the spatial distribution of those cells, the XY coordinate for every individual cell in the sample, so you can begin to study things like spatial relationships, spatial neighborhoods, and look at different cell types that are interacting with each other. Really high level, the way that we generate these multiplex images is through an iterative process of cyclic staining and imaging, which is facilitated by the sample chip itself. So the sample is loaded onto a standard 1 milliliter histology slide. You affix a cover glass on top that generates a microfluidic chamber. And through this microfluidic chamber, we can iteratively deliver reagents to the sample. We're doing antibody-based detection using primary antibodies that are directly conjugated to common non-proprietary fluorophores. And we can do up to 5 per cycle. And then we stain. So after staining for those 5 antibodies, we collect the image data on those 5 targets. And then we remove the fluorescent signal primarily by photo quenching the fluorophores. So the antibodies remain bound to their epitopes, just no longer fluorescing anymore. So then we can come back in again using the automated fluidics and stain for with another cocktail of up to 5 antibodies, 5 new antibodies to 5 new targets, reusing those same 5 fluorophores in the previous cycle. And we just do this over and over again to build up a high plex assay, 5 targets at a time. Today, I'm going to talk about 4 main benefits of the CellScape platform. We're really interested in quantitative performance, making sure that we're not just measuring the presence of or absence of a protein, but really quantifying protein expression across the full continuum of expression range. We're really proud of the kind of straightforward and reliable chemistry that we're able to deploy, which gives users really robust performance and users are able to get to productive results much quicker and at a much more scalable cost and throughput and plex. Also, because of the kind of straightforward and gentle chemistry that we're deploying, we have this sort of feature of modularity at any time, the ability to store and probe samples over and over again across many days, months, and years. So we'll talk about that. And then really with this simple chemistry, we get quite a bit of flexibility, really designed with kind of an open platform in mind that allows customers to kind of expand the utility of the box and support other types of less conventional assays. So these are the four main points that I want to talk through, and we'll go through them kind of one by one, starting with quantitative performance. So what is quantitative spatial phenotyping? This really comes from three different specific attributes. One is really high resolution. The second is really high dynamic range. And the third is across the whole slide. And so then you're really able to look at true single cell resolution. You're able to quantify protein expression across the entire continuum of expression range. And you can do this without having to select for certain regions of interest or anything like that. So resolution, I think this is pretty straightforward. With the CellScape, we're generating images at 182 nanometers per pixel. This is sort of best-in-class resolution and really allows us to look at not just single cell, but really subcellular features. Like in this example here, you can resolve that there's individual puncta of FOXP3 expression, for example, in this immune cell. But where this really comes into play is when we start thinking about things like cell segmentation. So in this example here, we've applied the exact same cell segmentation algorithm to two images that only differ in terms of their resolution. So on the left, there's a high resolution 182 nanometer per pixel image, and on the right is a more conventional 500 nanometer per pixel image. And you can just sort of see qualitatively that the cell segmentation algorithm performs better when you have a higher resolution image. But we can also begin to quantify the performance of the cell segmentation algorithm by looking at a number of different metrics, like, for example, nuclear area. We get on average smaller nuclei when you have a higher resolution image. You get nuclei that have more complex and more eccentricity in their shape. And this is, of course, more representative of what we see in biology and indicative that the cell segmentation algorithm is performing better. Again, we're applying the same exact cell segmentation algorithm to two different images with two different resolution, pixel sizes. And the reason that this is really important is that cell segmentation is the very first step in your analysis pipeline. So if there are errors in your cell segmentation, then they will kind of permeate all the way through all of your downstream analysis. So this really highlights sort of the importance of resolution in your multiplex images. We combine this high resolution image with a proprietary high dynamic range image acquisition pipeline. And in order to describe this, I think it's helpful to just define dynamic range in its most simple terms. And this is for any sort of quantitative assay. You're trying to correlate some signal intensity to some abundance of analyte that you're interested in measuring. And you're going to get a signal response curve that looks something like this. And your dynamic range is defined as this range in the center of your signal response curve where you have a linear response between the abundance of analyte and the signal intensity. And it's really important that if you want to have a truly quantitative assay, then the dynamic range of your assay, this linear detection range here, must exceed the range of whatever abundance of analyte you're interested in measuring. But in the case of protein expression, we know that the range of protein expression, the range between the lowest expressors and the highest expressors exceeds the dynamic range of the best camera that you can buy. And so the question is, how do we quantitatively interrogate proteins then using imaging, using cameras? And the way that we do this with the CellScape is that we actually take the same image multiple times using multiple different exposure times. And this effectively extends the overall dynamic range because we can quantify the really bright signals using really short exposure times, and we can quantify the really dim signals using long exposure times and sum these all together to create a new composite image with effectively a broader dynamic range. So what does this look like in a real biological sample? Here we are looking at, again, the same image taken multiple times using increasing exposure time. And I'm displaying here just a single marker, but we're showing it as a gradient display so that you can really see the variation in intensities in pixels. And whenever you see red in the image, those are saturated pixels. And we've highlighted two cells. We have a high intensity cell highlighted by this circle, and we have a relatively low intensity cell that's highlighted by this square. And what you'll see is that these lower, shorter exposure times, we can nicely detect and quantify this high intensity cell. But you don't see much, if any, signal from these low intensity cells. As you increase the exposure time, you start to detect this lower intensity cell, but you start to saturate some of the signal on the high intensity cell. And what that means, again, if you have saturated pixels, you can detect that pixel is there and the protein is there, but you can't really quantify how high the expression is because the detector is saturated. And the main takeaway from this is that there is no one single exposure that is optimized for quantifying the expression of protein on both the high expressor and low expressor cell simultaneously. So you really do need multiple exposures to quantify across this full continuum. And then what we do is we take signal intensities on a pixel by pixel basis from each of these images and sum them together into a new composite image with a broader range of pixel intensity values. So that's the why. If we look at a really specific example, this is a user who was able to, in their validation data, they were able to discern using cell lines high, intermediate, and low expression, which correlated to their ground truth Western blot data. And so what this shows you is, again, there isn't just a positive and negative phenotype, but there can be phenotypes that exist across the full continuum of expression range. And because of the high dynamic range image acquisition with CellScape, they were able to replicate this in their cell lines and then see this again in biological samples, again, looking at a full kind of continuum of expression ranges. This high resolution and high dynamic range imaging is combined with a really large imaging area, which means that you as a user, when you're designing your experiment, you don't have to bias your experiment with looking at specific regions, or you can even fit multiple samples within our 44 by 16 millimeter imaging area, which allows you to do really quantitative comparisons between samples. We've also done things like TMAs and really large blocks that you could fit within this imaging area. So to sum up, when we're talking about quantitative spatial phenotyping, we're really talking about combining best-in-class resolution imaging over a really high dynamic range that gives us a quantitative detection across both low, intermediate, and high expression. And we're able to do this across the entire slide using standard 1 millimeter histology slides. Switching gears slightly away from a little bit of how we collect the images to more of the chemistry that we're deploying for protein detection. And for this, I think it's helpful to contextualize with some of the other approaches that are available on the market. Almost all spatial proteomics platforms are using antibody-based detection, and they just vary based on what the antibodies are conjugated to. One common approach is using indirect immunofluorescence, so fluorescently tagged species targeting secondaries, and then getting these to recognize unconjugated primaries. One of the biggest challenges with this, of course, is a very conventional approach for low-plex immunofluorescence imaging. But once you start trying to multiplex at a really high plex level, you realize that it's imperative that you remove the entire primary-secondary complex. If there's any secondary remaining between cycles, or even any primary, unconjugated primary, this will show up as unspecific signal due to cross-reactivity in subsequent cycles. And so in order to get around this problem, you have to expose your sample to really harsh antibody stripping, which, of course, after a number of cycles has negative consequences on epitope stability and also tissue morphology as a whole. Another approach is using oligotagged antibodies. So these are antibodies that are conjugated to proprietary oligos, and then you cycle in complementary oligostrands, which are conjugated to fluorophores. And this leads to some challenges with antibody selection because oligoconjugated antibodies are sort of only available from proprietary sources. And we know that the conjugation chemistry that's deployed, at least on the most common commercially available competitors, impact binding affinity of the antibody and binding specificity. And so overall, this adds significant time to assay development and validation, because you may have an antibody that works really well when it's unconjugated. When you conjugate it to one of the proprietary oligos, that can impact the performance significantly. So whenever possible with the CellScape platform, we use direct immunofluorescence, antibodies that are directly conjugated to common and non-proprietary fluorophores. And this allows us to leverage really highly specific antibodies. We don't have to worry about extra steps with conjugation and assay validation that really increase the overall assay validation time. And it gives us a mechanism to remove the signal between cycles in a really non-destructive manner, which means that as a user, you can get to a much higher plex if necessary. We do this in a really sort of open source way. We do have a catalog of what we call VistaPlex assay kits, which I'll talk about in a minute. These are highly validated panels of antibodies for kind of core immune profiling and tissue architecture and immune cell subtyping applications. We also have lists of over 350 compatible antibodies, which can be sourced from other commercial antibody vendors. Or as a user, you can go source your antibodies from any other commercial antibody vendor, leveraging experience you might have with specific clones. Any sort of primary monoclonal antibody that's conjugated to a compatible fluorophore is compatible with the platform. And this leads to really fast and straightforward assay validation and customization. I'd like to also just take a quick moment to comment on our assay validation process. We invest a lot of time and effort in ensuring that our assay kits that we sell are really high performing, particularly when it comes to reproducibility. So, this is not meant to be an exhaustive description of our assay validation process, but just to say that this is something we spend a lot of time and effort and energy on, looking at multiple types of samples, multiple biological replicates, and scanning these across multiple instruments that are across multiple geographical locations, and ensuring that we have consistent performance when doing so, so that when you get these kits in your hands, you can have confidence that they will perform. Another important point is, the mechanism of signal removal between cycles. Again, all of these highly multiplex immunofluorescence, spatial proteomics platforms are using some cyclic process of staining and imaging, and inherent to that, there's some signal removal step. And what's important to recognize is that not all signal removal methods are created equal. Some platforms using secondary antibody labeling, really require harsh antibody stripping methods that are going to damage tissue and epitopes. There's also other photobleaching approaches that expose the sample to kind of the full spectrum of light. And with CellScape, we use what we call filtered photobleaching, where we're filtering out the UV and the IR wavelengths to quench the fluorescent signal with the absolute minimum impact on tissue and epitopes. So what does this look like? Just sort of selecting a few examples here, looking at simulated photobleachings, something to replicate something like 20 rounds of photobleaching. You can see a really consistent signal from kind of the pre-photobleaching to the post-photobleaching rounds across, essentially all targets of interest. If we compare this to elution or stripping based methods where you need to remove the primary secondary antibody complex, we know that we see really significant impact in not just the epitopes, but also the tissue morphology. You can see sort of a complete withering of this sample here, just looking at H&E after simulated 20 elution rounds. So our approach allows us to get to really high plex if necessary, up to, in this case, we did 100 markers. Again, really high plex with this quantitation across the continuum of cell types and expression values. Switching gears to talk a little bit about the modularity piece, I think this is one thing that is enabled by our gentle chemistry. But really, there's a number of kind of complaints that are associated with these high-plex spatial proteomics platforms that require you to design your assay up front. You sort of have one shot to get this experiment right, which may be a very expensive and time-consuming experiment, but you maybe don't know that your sample is low quality when you start out. Maybe you find after you run the experiment that your sample doesn't reflect the specific biology that you're studying. Or maybe your sample is good, but as you analyze those results, you realize post hoc that there are additional markers you want to probe, and so this can lead to kind of money and time and precious samples going to waste. Because we use, again, really gentle chemistry for all of the staining and image acquisition and signal removal, we have the ability to stop and store the sample and kind of come back to it at many points over the course of days and months and even as long as 2 years. This slide kind of shows you a number of different reasons why you might, as a researcher, leverage this feature. Again, kind of upfront, maybe you use this to assess suitability or quality of your samples up front. Just look at 3 or 4 or 5 markers to make sure that, if you're studying tertiary lymphoid structures, you want to make sure that the samples that you're looking at indeed have tertiary lymphoid structures in them, for example. So you could use this as sort of a cohort selection criteria. As you kind of progress in your experiment and iteratively stain and analyze the data, maybe you identify additional markers of interest over time, maybe even more longitudinally. More markers are discovered of interest. Maybe you develop new hypotheses. Maybe you want to conduct longitudinal studies. You can go back to the sample for up to 2 years and reinterrogate that exact same sample over and over again. So here's a really kind of specific use case of that. This was a grade 2 invasive ductal carcinoma, a breast carcinoma, which was initially stained with our core immune profiling panel. And researchers sort of had this initial observation that there's a really strong immune infiltrate that is accompanied by this kind of regional immune exclusion. And so this leads to this sort of obvious follow-up question of what is the mechanism of immune exclusion? And if you were using another kind of competing platform that doesn't have this reinterrogation capability, what you would need to do is you would need to go back to the block, cut another section, and restain this section with your initial kind of 15 or 20 markers, and then add on some additional markers on top of that to begin to test this question of what's the mechanism of immune exclusion. In our case, we could go back to the exact same tissue section. We could reinterrogate that same exact section with additional targets and answer that question right away. And we can see that this immune infiltrate is compartmentalized by the CD138 plasma cells and epithelia, which may act as sort of an immune barrier, which gives us this kind of ability to conclude that the spatial arrangement within the tumor is impacted by these plasma cells and epithelia, and maybe has some implication for the course of development for treatment. And we can just keep going with the same section over and over again, reinterrogating and reanalyzing. And this is a really nice section because you can see sort of the progression of the disease across the specimen. And what this feature, this ability to reinterrogate over time, what it allows you to do is really get the whole story from every sample and not worry about kind of nailing that perfect experiment in the first shot. And I think it really is sort of a feature that can allow you to rethink the way you design these experiments and the way you sort of can future-proof your studies leveraging this reinterrogation capability. So that's I will conclude there. I thank you for your time. Just to summarize, the CellScape spatial proteomics platform with a real emphasis on quantitative spatial phenotyping, not just detecting the presence or absence of protein targets, but really quantifying across the full range, the full continuum of protein expression in biology. We're doing this leveraging straightforward and reliable techniques that give us a lot of flexibility and the ability to have modularity in our assay design over time. So once again, thank you for your time, and I hope you enjoy the rest of the program.